like the very difficult problem that Alok was talking about, I'll give you one which actually is soluble. And this is one where we are talking about a particular kind of an eye disease. In India, something like about 33 million people are visually impaired. Out of that, about 11% suffer from glaucoma. The eye is a fluid-filled ball. So the pressure that you see inside the eye because of the various fluids in there is approximately anywhere from 5 to about 15 millimeters of mercury above NTP. If it goes above, there's not enough space for the eye to expand. So the optic nerve, which is in the back, gets hit. And over time, the optic nerve loses its function and it causes visual disparity. That's called glaucoma. We also see, just like what you pointed out, the number of childhood or congenital cases of glaucoma in India is substantially large. That's referred to as primary congenital glaucoma. It's a severe form of childhood blindness where you end up getting various kinds of defects of the trabecular meshwork, which is the meshwork that's surrounding the anterior part of the eye and the anterior chamber angle through which the aqueous humor actually moves around, circulates. And you notice one of the things that was done by our institute was looking at the prevalence of various kinds of ocular diseases. And I'll just talk about this particular one where you notice the prevalence of PCG in Western countries is somewhat like 1 in about 10,000 to 30,000. In contrast, you notice what is happening in India. It's something like 1 in 3,300. 1 in every 3,300 children born may suffer from primary congenital glaucoma. Also look at, for interest's sake, I'm putting also the Slovak gypsies who seem to have historically moved from certain parts of India to go on to Eastern Europe, particularly the Slovakian countries and so on. So it's clear that there is a genetic basis behind this. And what are the various kinds of genes associated with it? There's been some work done by a variety of people, and at least there are something like about 5 of those which are known. One of which I'll concentrate is one called CYP1B1. This has to do with the gene that really is a cytochrome P450, which is a membrane-bound protein that transports a variety of things and so on. I said PCG alone results in 4.2% of blindness in India. The clinician can interfere and restore vision. I'll tell you how that is done. And when we looked at in India alone, notice in the case of variety of populations within India, in the south, it turns out to be we looked at about 212 patients, east, west, and north, and we noticed clearly CYP1B1 is present in 43% of the children that we looked at. And 35 mutations in the CYP1B1 have been observed, and the most common was essentially arthritic. The other interesting thing also is 42% homozygous. So that's an important point also we need to look at. As a biophysical chemist, I worked along looking at the structure with a molecular dynamics person, Nagaraja Ram, who used to be first in CDFD, now in the University of Hyderabad. We worked together to look at the structure in solution and also the model in here. And we are pointing out what are the various mutations out of the 35 that are put in here. What are the structural properties of disease mutants or molecular phenotypes of proteins as compared to their wild-type counterpart? As I said, CYP1B1 is a cell membrane anchored protein and it's involved in the development of the trabecular meshwork in the eyes. And that's where the difficulty is. Certain mutants have been found in patients suffering from a genetic disorder called PCG, and the mutants that we were able to look at in the biophysical part are all these 6 of them, and those are the papers that I'm pointing out. And the properties of the functionally important mutants also we looked at. When you look at, for example, the substrate binding region, which is in here, one notices that all these mutations that we see in here, A115, 132R, and so forth, and the most predominant one, which is G466D, the substrate binding regions that we in these mutants are too narrow or too wide, so they don't fit very well. So clearly there is a molecular fitting that seems to be a problem in here. And likewise, not only a binding region, but is there even an access to it? And when you looked at these, we again find that substrate access varies also widely. So there is no tight fit that's possible in the mutants as well. These are some of the insight that we end up getting by just looking at the molecular structure of the protein. About the same time, Yashoda Ganekar, who used to be with us at that time, worked along and looked at the polymorphism, the promoter function. So not only do you have a problem with the gene, but even the promoter region seems to have difficulty in here. When you followed it through for the CPON-B1 assay, we found that the survival rate goes in the essentially as a function of months or so. So you have a problem here with respect to the protein itself. What is the kind of frequency that we see? We said it's when you looked at comparison of what's seen here with elsewhere in the world, one notices all the way from Australia down to US and Brazil, it's widely prevalent across the globe. That's an interesting part, because how would it have been all across the globe and so on? Furthermore, if you look at that, you notice a variety of heterozygous mutations, homozygous mutations, as well as compound heterozygous mutations were seen in here. And of these, the cases without CPON-B1, in other words, other genes may be responsible besides CPON-B1 alone, widely prevalent across the globe. So, if it is widely prevalent across the globe, can we now start doing a haplotype analysis? It's a group of sequence variations and repeat sequences that you notice in tandem in the DNA sequence. That was done on Indian patients first versus Indian normals, and they were compared to those found elsewhere from available data, Brazil, Australia, the Slovak gypsies, and so on, and looked at what would be the various possibilities. I won't bore you with all the big details in here, essentially to point out that the haplotype that you see is prevalent all the way from Ecuador down to Brazil, and of course India is right in here. And you notice specific mutations are seen in Saudi Arabia, India, Brazil, and so on. This particular one, called the CGCCA and CGGTA, are both very common. So there is a common origin that we find in here. And if you look at what the origins are and what the mutations that we see, what's the prevalence of those mutations in all these countries, you can actually start building an ancestral map, something that was done by Partha Majumdar and Shubha Chakrabarti in our lab. And it also turned out rather interestingly, about the same time, we found that monkeys, particularly chimpanzees, also suffer from primary congenital glaucoma. And so we start looking at the haplotype in the chimpanzee and how it would have moved from the ancestral. With just one recombination, you can generate the entire sequence as well as the haplotype of the PCG gene. So movement, you can see globally, CPON-B1 mutations in PCG are strongly structured by geographic and haplotype analysis. Work was done together with Partha Majumdar in here. And that's the one I am talking about, you notice. And so, what would have been the genetic determinant was a question that was asked. And he pointed out gypsy genes may tell us the history about these, largely because people believe some of the gypsies were actually of Indian origin and so on. And a lady called Luda in Western Australia had been doing some work on gypsy genome and also looking at the gypsy gene PC, primary congenital glaucoma. And their conceptual framework at that time was we'll start looking at isolated founder populations. And the Balkan gypsies are a particularly interesting idea, plus of course the Indians that we find. So isolated founder populations drawn from large ancestral populations on one hand, with the gypsy that are seen in the entire Bulgarian region, and of course compare them with Indians and so on. And the recent founding in evolutionary time scale, no more than a few thousand years. And the ancestral disease causing allele on a same haplotype. The haplotype sharing was due to less number of founding populations. And further validation of this came out in outbred populations from Indians, Australians, Africans, and Brazilians. We did that work. And when we found that not only CYP1B1, but another gene, this is the latent transcription binding protein B2, that mutation is also associated with ocular phenotypes both in Roma gypsy populations and Indian populations. That's your haplotype that you can see in here. And you notice the percentage of these essentially are compared to each other. So we notice that this is the kind of a map that you end up getting for what would have been the possible origin, ancestral origin of primary congenital glaucoma. So the conclusions, if that is so, you should also start looking at not only haplotypes, is it even possible for it to look at what is called the ROH or the run of homozygosity in these cases and so on. A harboring G2 could be potential associated with genes that needs further replication. And the founder mutation in gypsies and absence in Indians indicated a rapid admixture and population stratification among them. Purely of genetic interest, of not necessarily. So we started looking at it. Should we start using this kind of a thing, using zebrafish, keep on knocking down one after the other the mutations and so on? And I'll give you the final one with PCG. So far, we were able to get CYP1B1 responsible to about 58% of the global, and of course Indian PCG. In addition, that other one, the lateral latent transcription binding factor on TIE. When you start doing a targeted sequence, then you find that reduces somewhat into 48%. And you do the whole exome sequencing finally, it becomes even further. And that means there are multiple genes associated besides CYP1B1 and so on. This is where the genetics part stands and the biochemistry part stands. Now, the conclusions for this would be that the exome sequencing indicates further genes involved that may be involved in PCG, and functionally, these may interact with the CYP1B1 and other genes through a common biochemical pathway. We need to look at the biochemical pathway now. And we are currently looking at both the LTBP2 and CYP1B1 in terms of molecular structures and so forth. The synergistic effects of these genes may hold to the PCG pathogenesis. So we now understand a little bit about the biochemistry, the genetic prevalence, and the genetic ancestral reasons and so on. How do you treat the disease? This is where the clinician comes in. So you need to have an integrated management. I started with my title as translation. What one means in this case of translation is bench to bedside. What do you see in the lab bench? Does it apply to the bedside of the patient or bedside to bench, whichever way you want to have a look at, and then on to the community? And that requires not just omics and biochemistry, but also requires medical, surgical treatment. There are no easy medical treatments other than just want to dilate a little bit more and so on, but that's a temporary situation. Surgery can provide you, and in cases where it is not possible to do rehabilitation, Here is an example of one such. The clinician that we have, Dr. Anil Mandal, from 1991 on, must have easily treated close to 1,200 children. That's a remarkable thing that he has done, because he was one of the earliest people to get the Bhatnagar Prize in ophthalmology. That's how the baby is seen when it first comes to the hospital. After the trabeculectomy, where you actually open up a little bit so that you dilate the area and so on, you can see clearance happens. 6 months later, the baby still needs to wear glasses. He has followed this child all the way up to 25 years. He has been able to do this for close to about 80 such children, and been able to look at what would be their profession, what would be their eye health, and so forth. And the last one is a marriage that we actually attended. This is the guy, this is the doctor who actually treated this patient and so on. What I am summarizing is essentially one coming together of genetics, basic science, treatment modalities, and rehabilitation wherever possible, and so on. And that, I think, is an example of a translational research that I wanted to point out. Thank you.